Hi, my name is Lucy, and today we're going to introduce you a tissue filter and sterile technique. First of all, you have to wash your hands, ethanol the hands later on, use a clean wipe, and then now we're going to ethanol again our gloves thoroughly so that we don't contaminate our working area. Let your hands dry, and before you do anything, we have to set up our station. So I arrange my materials in a semicircle so I can have this space clear. I don't have my hand passing over any of my cells, contaminating them. And then I put all the materials, I feel more comfortable, of course, on the right side to have my tips and my pipettes, and then the toxic and liquid waste on the back. Okay, so now we're gonna take our cells from the incubator. I'm going to observe that they're in contact.
So what are you looking for right now?
I'm looking for cells to be at the bottom of the flask. I decided to have a 90% confluent cells, meaning that they're going to cover 90% of the bottom of the flask. So as you can see, these are CHO cells, and they're 90% confluent, and they're attached to the bottom of the flask. You can see that they have kind of like arm-like structures in order to attach. Now that we confirmed that the cells are ready, we're gonna first ethanol our clean wipe, and we're gonna clean the flask before introducing it into the filter. After that, you also want to ethanol your hands again. Rub them. And let it dry. The first step for trypsinizing cells is to take out the medium. So we have our liquid waste here.
We're taking out just to get rid of the FBS.
That's right. Before we use the trypsin, we have to take out the media first. The media has a mixture of antibodies and also FBS, which is fetal bovine serum, and that actually blocks what the trypsin does. So even if you're adding, if you don't clean it nicely, the trypsin won't work. So we introduce our serological pipette. We go to a corner so we don't touch the surface of the flask, and we carefully extract the media. After you're done, again, you see how I'm using my two thumbs to close? You want to always do that. The flask is done, push it to the back so you still have your free space. And then we toss out the media.
And so you're kind of working in a semicircle.
Yes, that's right.
Middle.
Everything again is in a semicircle. Okay? After I'm done with that, I want to rinse it too to make sure that there's no FBS left. So I use a sterile PBS at room temperature.
And PBS is just a fluid that simulates the cell culture but doesn't have any of the enzymes.
Yes, it's a phosphate buffer saline solution.
So it's just at 1%?
Mm-hmm.
Cool.
And again, I'm using my two fingers and I'm just extracting here the PBS. And again, I don't go over the top, I just close it and again push it to the back to keep my area clear. I bring the flask and I open the flask, carefully add the PBS. Not too fast because you don't want to lift the cells from the bottom. You want to be gentle.
So you're kind of just washing the cells, but
yes,
knowing that they're still attached to the plate.
Correct, that's right. And then I'll do a couple of swirls to make sure that the PBS goes to every single corner and it covers the cells evenly. After I'm done, I have to take out the PBS. So I go again and get a new clean pipette. And again, I go to a corner of the flask and I gently remove the PBS. Make sure that you take out everything. And then you discard.
Can we use some of the pipetters again, or would you prefer to get rid of them all the time?
It's better to get rid of them all the time because it keeps the container sterile. If you keep, especially I touched the bottle, it's already a toxic waste, so there's a lot of contamination and you don't want to introduce that again into your flask with cells or anything that has chemicals or any PBS that you're using.
So would a good rule of thumb be
It's
if
always,
you think you touched something, just go ahead and use a new...
Yes, and even if you're doubting, it's good to still change it. Okay, so that's it helps a lot. So now we're gonna use the trypsin to detach the cells from the bottom of the flask.
I noticed that when you were pouring the trypsin, you kind of made it flow all over the cells.
That's right, so that I can cover the surface of the flask. So I'm gonna have to let the trypsin work, and basically what it's gonna do is it's gonna start kind of like degrading the proteins that the cells uses to attach to the bottom of the flask, and that takes around 3 minutes. So we're gonna wait. And we also have to go back into the microscope and check how they look and make sure that they are detached. After a period of 3 minutes, we check under the microscope and we can see that the cells are now round shape. And if you move the flask, they're already flowing, meaning that they're already detached. So now we're going to proceed to stop the trypsin by adding media. After I check my cells, again I ethanol my flask to keep the station sterile and also my hands. Now I'm gonna add my media to stop the trypsin. And again I'm using my 2 fingers and then making sure that I don't pass my hand over any of my containers.
So basically here you're adding the FBS to...
Yeah, the media that has the FBS.
And it deactivates the trypsin.
That's right. So the cells will not degrade after a period of time. After you're done with that, we're going to transfer the cells into a 50 ml conical tube that is already been labeled with the name of the sample, my initials, and the date. It's always good to keep track of your samples by doing this. So I can use the same pipette, and then I'm gonna collect the cells. Put my flask in the back, and again I keep my station clear. Discard the tip.